Thank you. Thank you very much for the invite. I'm really excited to hear about the great, developments lately. I've been here at EMSL quite a while ago, but it's amazing what new technologies are coming online. So thanks again for the invitation to talk about our very recent project. So this was funded a year ago. All right, so this is the multi-modal imaging to understand basically the plant-microbe interaction. So it's a new project, as I said. It's a BR project. And then there are many people on the project, so I will only be able to talk a certain slice of the overall project. We heard many talks about the importance of that plant-microbe interactions, how the microbes are supporting plant growth and health, and providing, nutrients, protecting against pathogenic microbes, et cetera, et cetera. Then what you see here on the right is the like a plate grown in Gene Greenberg's lab, one of our collaborators, where you have Arabidopsis grown without and with Pseudomonas simi, and you see, a really different morphology, root morphology, as well as an increased aerial tissue. So, as I said, I'm really impressed by the different technologies of being able to look at thousands of metabolites, mapping them spatially onto the sample, being able to do omics, proteomics, transcriptomics. So it's really amazing. But this project is sort of going back in time a little bit. And what we are trying to do is look at more spatiotemporal changes during that root colonization. So for this, what we are doing is to convert some of these plant growth promoting microbes into biosensor cells. So with that, we'll be able to see chemical exchanges between colonization, as I said, and we can follow that colonization for the first couple of weeks. So the goal here is to develop these microbial cells, also develop a high throughput imaging capability, so some automation in there, and then also include AI and analysis of that data. So the group is composed of Yasuo Yoshikinu's lab at JGI, providing synthetic biology capabilities, Gene Greenberg, the plant science end of things. And then from Argonne, we have people from biosciences, data science learning, as well as for X-ray science division. So we have Jessica, Johnson, Chase, Ken Kemner is here, Rose Wilton, and then you will hear from Neil and Pham Phong. So they are the AI component, and then Si Chen was not able to present, but she's working with Olga, so she's providing APS capabilities. Good thing is we have Ken, who is providing APS expertise as well as the biological expertise. So we used, live systems as sensors. We know very well that, for example, in humans, we have a large dynamic range of sensing. So we can sense chemicals that are in the femtomolar range up to millimolar, for example, glucose versus insulin. Bacteria probably doesn't have that same dynamic range, maybe 10 to the 4th or so. But for example, E. coli can sense 3 molecules when it comes to chemotaxis. Obviously, this is not a transcription factor-based system. And then so the goal here is to couple some of these transcription factors to a reporter, fluorescent reporter, and then either working with an active or derepressed promoter, or maybe even engineering a NOT gate to to turn downregulation into upregulation of the signal. And then we'll then characterize these for dynamic as well as operational range. Very similar to previous work we've done in the lab as part of Ken Kemner's led Small World Bioimaging project, where biosensors were developed in a bee subtilis backbone against xylose or reactive oxygen species. So we'll basically apply a DBTL approach. We are selecting some of these transcription factors. We have an experimental transcription factor discovery pipeline in Yosu's lab. I'm not going to talk about those, but then once we identify these, we characterize them in vitro and then also in microfluidics chips or plates, as well as applying an orthogonal approach to look at elemental distribution in their interface. And as I said, there's an AI component to make sense of the data. So I'm going to present some of these. So in previous projects, we engineered or maybe over-engineered a single organism to be able to sense the given analyte. This time around, last fall, we went to 70 different locations and isolated microbes from poplar rhizosphere. And Jessica was able to morphologically cluster them into more than 40 clusters, and then we sequence these. So at this point now we have a full genome sequence with nanopore, as well as some of these also have Illumina polishing. So we have a fine print of what's capable. And then so most of the characterization work then was done by Jessica and Chase in this project. Just giving you an overall view of the current status of the project. So I'm not expecting to read everything, just look at things that are in green. So these are some of the organisms that we isolated, and also we have organisms that were provided by JGI and UChicago. We looked at antibiotic resistance. We looked at where we can express easily fluorescent proteins if we can. Then we also did biolog phenotyping on these organisms. And then since we had labeled cells, we also looked at colonization in Arabidopsis, Camelina, as well as poplar. And then we also looked at whether they can produce IAA, whether they can also solubilize phosphorus, can they form biofilm, as well as see if they can survive on azelaic acid. So this is just a snapshot of a 2D cluster. And then if you look at it, so for example, on this side, we have sugars. On the right side, we have small organic acids that are normally in the exudates. And then you can see that you can find an organism in our repertoire that would respond well to one of these metabolites. So then it's a prime candidate for turning that into a biosensor. So as I said in previous work, with Ken, we developed a microfluidic approach to monitor root colonization. And then we could actually see forming these colonies, sometimes even dissolving those colonies and then relocating them somewhere else. This was a very interesting story where we also found some differences in terms of the colonization pattern. So that was very important for us. And then these were the sensor cells where we could turn green fluorescent protein production upon colonization for xylose or reactive oxygen species detection. And then, as I said, we collaborate with Jean Greenberg's lab, and she's been kind of working on figuring out a pathway that leads to, helps in the colonization signals. So, for example, as Arabidopsis root senses PGPRs, there are some signals. It has a transient low iron signal that a cascade is initiated. This is more like a kinase cascade. And then as a result of that, we have an increased oxidation of sugars, amino acid, or azelaic acid, etc. And then this will then sculpt that microbial community around, which in turn will help the plant. And the nice thing is that both Yasuo and Jean, they're working on the same plant system and also working on the same microbes, so Arabidopsis Pseudomonas simi. This is just a snapshot of one of the biosensors. So as I said, plants will also secrete or excrete azelaic acid, and this in turn will promote growth microbes to grow, and then also somewhat limits pathogenic microbial growth. So this was one of the important metabolites. So here is azelaic acid, C9, and from a recent paper, they were identifying the potential transcription factor. We simply clone that with the promoter region and turn that into a biosensor. So these are the microbes that can grow on that azelaic acid, and this is a biolog phenotyping result where on the x-axis we see respiration of the microbes, and on the y-axis we see green fluorescent protein signals. So as you can see, most of the chemicals didn't elicit any fluorescence, but the C10 version of the sebacic acid clearly elicited a response. Just another example where we have lactic acid. So this was a native promoter. We just inserted into that organism, and then so it will respond on lactic acid as well as glycolic acid. And then on the right, we have another example. This is one of the sensors that Yasuo's lab has developed. What's unique about those, and I'm not going to talk about it, but they basically expressing a promoter library that was inserted into a landing page, landing pad with GFP signal, and then they were able to look at by transcriptomics to see what those promoters were activated during colonization. And then so they then identified those that were up or down regulated and then turned them into GFP linked reporters. So this is one of those that promoter is around this protein that's involved in APS synthesis. Here's just an example of 3 different microbes, 3 different plants. So Arabidopsis, poplar, and then Camelina, how they were able to colonize. And then we're using, in this case, a GFP variant that Rose Wilton has developed in part of the Small World project. And it's a stable label that lingers around without selection for weeks at a time. We also wanted to take advantage of the APS capabilities before the shutdown. So what you see here, here we have the GFP labeled bacteria, Pseudomonas sbw25. And here gold labeled on the surface, we showed in the confocal that the cells were still kicking. And then this is the result at the bionanoprobe by Chen and her postdoc. This is the tychography image. On the bottom left, you see the bacterial cell, and you see a fairly large bright spot on it. And then when you look at elemental distribution at 50 nanometer resolution, you can see in one of the panels here, so calcium was lighting up the microbe, and then you can also see that gold label on that. There is some interference with other elements, but it is the gold label that you see. And then, so we use this approach, and now we also labeled the plant side of things with just rhododextran, so that I could see under confocal the plant and also the labeled microbes that were interacting with that plant and colonizing. And here we used quantum dots actually on in this experiment, something that Ken Kemner is an expert in providing that capability. And then if you look at in this elemental analysis, so what's different here is that the quantum dot is labeling the plant side in addition to the rhododextran. And then we still have the gold labeled microbe. So you can see that here's the gold label. So it's on the surface. And then you see the cadmium from the quantum dot to highlighting plant features. But obviously there are better elements that will provide a nicer image, such as calcium. So we also wanted to take advantage of this new entity, Eberlite. And so we were able to do experiments as one of the pilots, part of that. So this is the Eberlite, an APS user program that is helping BR community science, and also now part of the FICUS program. And it was mentioned before, Olga and others, that we have capabilities of sampling different environments, so atmosphere, root, plant, microorganisms, as well as looking at different scales. So this is very similar to what was said in the previous talks. So this is a new entity, and we are very excited to be able to provide BR users multi-beamline experiment, also organizing how that sample travels through that multi-beamline experiment, destructive at the end, et cetera, et cetera. So we're excited, and you will hear more about it in publications. So this is the experiment. This was a micro CT experiment. So we had collected the poplar root, right? And then so we were able to, we had the root, and so we could just use micro CT to look at root and the soil aggregates that was on the root. So this is one of those images. You see the X, Y, and Z. So this is the reconstructed Z slice of that interaction. So you see the root as well as the soil aggregates around. So we collected many images before the shutdown, and that also enabled us to work on the AI aspect of things. So what you see on the top left, this was AI guided image analysis, simply thresholding the image, providing edge detection, and then later on identifying those objects, and then in 2D. And later on, these were then reconstructed in 3D into different objects. So those are the particles. So now we have, in just one of the 2 millimeter slices of the root, we have many objects that were then combined into less than 6,000 volumes. Neil in Fanfang's group also looked at deep learning approaches. So this is a different approach using Max RCN, where he could not only detect objects, but maybe categorizing them also, which will be also important down. And then just for looking at if language models could be used, you can actually apply a language model that identifies an object, a round object with a hole in it. So this is what ChatGPT-like language models can provide. So in conclusion, I We have a fairly complex project. It has microfluidics microscopy, sensors, synthetic biology, EIML, and also an orthogonal characterization by X-ray. I didn't talk about the automation aspects, but this is basically the overall project, and I'm very excited that maybe being able to use EMSL capabilities to actually define some elemental analysis or metabolite mapping, et cetera, et cetera. And then so I'd like to thank the team members. As I said, right now I only talked about projects that were worked on by Jess and Chase, and then I had Ken's contribution, Rose's, and as well as the She's, and then as well as the EIML people. And I also would like to thank the BCIS funding for this, and thanks for your attention.
Thank you, Gyorgy. We have time for a few questions, if you guys have any.
Hi, thanks for the talk. I really enjoyed the biosensor work. I was wondering, if you have tried to see whether or not those biosensors are at all quantitative for those metabolites? Like, will they fluoresce more if there's more azelaic acid, and can you quantify that at all?
Yeah, so we are in the process of characterizing that, but yeah, to your point, that will be the idea, is to see if they graded approach with the xylose sensor we characterized, and we could see that graded approach. And the idea here is to have not only that you can combine, so you can multiplex maybe 10 of these isolates, they respond to different things, as well as different colors, so we can see graded response, spatial, temporal changes during that early colonization.
Nice. Have you thought about validating those with the like chemical imaging or anything to see if that's actually the metabolite that's making it light up?
Right. So or only, right now we are just using the biolog, phenotype arrays, but would be nice to have EMSL capabilities to map those and to image those into the high. Your chip is nicely characterized.
Thank you.
I have a question. Can you give an example of the AI-guided segmentation for tomography? Was it applied to a specific project?
Well, this is the project. So they're part of this BR project. There's the AIML, and then since they are going to look at root morphology, etc., but we didn't have images, so we quickly collected CT images, micro CT images, and then so they already have done some nice AI-based methods. So something that might be good to talk about with you, Tamás, to see what segmentation approaches.
Is it like using a bunch of root image training data to help the segmentation of another sample?
Right. So the one I showed, just the regular segmentation on the left. So that was just thresholding and looking at and recombining 2D images or 2D slices. In the RCN approach, there's actually built like a little training model. It was existing models, but then he was able to click and then with that kind of maybe even categorizing some of those, big, white, big, whatever. So you can actually detect the objects just like other CNN methods.
Thank you. It's very exciting to see what's happening. I do segmentation myself, so I just wanted to mention that It's very hard to beat stuff that's being done in Facebook. So segment anything is a great model. So if anybody wants to do anything, that would be, if you want something out of the box, that's a great tool to use. For scientific image data, it doesn't always work though, which is good because it keeps me in business.
Right.
But within the ALS, there is a BES funded program going on. It's called ML Exchange. And as part of this approach, we are developing web-based applications for segmentation, so people can upload their data, or they're automatically uploaded to a web server using Plotly Dash technology to hand annotate and then have in the background automatically spawn segmentation models running. So that stuff is there, almost available. And if anybody's interested, just pull my nose or ear, or whatever you're comfortable with.
Okay, thank you. I will pull your whatever. Yeah.
Okay, let's thank our speaker one more time. And thank you to all the speakers in this session.